Today we are going to discuss rational design in medicinal chemistry, and mostly we are going to discuss the concept of QSAR and QSPR. In the next lesson, which will be tomorrow, we are going to have practice related to establishing QSPRs on some data sets, some of which we have discussed previously, and some will be new. So before going into what QSAR is, let's discuss why should we rationalize drug design. So I've shown you this figure, which is the famous funnel diagram, and it illustrates the common problem in drug development. So if we consider, if we have one approved drug, in the end, usually the base of this funnel is really large. So basically, in many drug development processes, people have about at least thousands or maybe even tens of thousands of the drug candidates that are predicted to be active during the basic research. However, there are many trials which filter out potential drug candidates because some of them may show toxicity, some of them may be inactive in in humans, for example. This process takes a lot of time and takes a lot of money. Sums like billions of euros or dollars are usual for a drug development process. So that's basically why we need to try to rationalize drug design. And of course, machine learning approaches that were established previously and that are developed today don't negate the need for clinical trials. So these are necessary stages. However, making the funnel smaller would be a great endeavor indeed, good aim. So there are two main paradigms in drug design. So the first one I would like to mention is the so-called structure-based drug design. So here we fit ligands to a known target, may it be some protein, for example, related to some disease. And the second one is the so-called ligand-based drug design. So in this paradigm, we screen potential ligands using available activity data. So we search new active compounds by filtering potential candidates from libraries of potentially active compounds. And classic approach of the first paradigm is molecular docking, which is basically which is based on molecular mechanics, force fields, and in this paradigm, interaction energies of potential ligands with targets are computed. We aren't going to discuss this paradigm because it mostly relies on machine learning, and there are some studies in which machine learning approaches are applied to docking, and in which machine learning approaches are introduced into structure-based drug design. However, these are mostly sophisticated ones. So we will focus today on the classic approach of QSAR and QSPR. So what is QSAR? QSPR basically is. These are abbreviations of quantitative structure activity relationships or quantitative structure property relationships. In this paradigm, we have some data set of potentially active chemical compounds, and some of them are we have a data set of active and inactive chemical compounds, and we want to build a model that predicts activity of new compounds that are similar to ones from the data set. To do this, we build some representations of these molecules, and in this stage, we need to predict structure of these compounds. We can use SMILES representations, or we can use molecular mechanics or quantum chemical methods to predict 3D structure of molecules in solution, for example. And taking these structures, we compute structural descriptors. Some of these descriptors we have discussed in previous lessons, and some of these descriptors we have calculated in the previous practical lesson. So next, researchers build machine learning models. These can be linear regressions, artificial neural networks, decision trees, whatever, to predict the property of interest based on these structural descriptors. So here we are in the paradigm of supervised machine learning, and this is a classical field. For those of you interested in this approach, I suggest this review, which was published quite recently by an international team of researchers. And I need to mention that, in fact, this approach is several decades, was proposed several decades ago, in fact. So the first one of the first and probably the first article that in which a quantitative structure activity relationship was built, formulated is this one in Nature. So in this article, researchers correlated some structural features of phenoxyacetic acids, so substituted. This is the molecule of unsubstituted phenoxyacetic acid, and we can introduce some substituents in various positions of the phenyl ring. So the authors proposed to use two simple descriptors. First, they proposed to use the previously mentioned partition coefficient, so between octanol and water. So we have discussed this descriptor. However, researchers used non-logarithmic form here, so they compared partition coefficient of substituted phenoxyacetic acids with the value for the unsubstituted acids, and also they used the ADMET parameter, ADMET constant, and correlated this with biological activity. So they obtained this equation, and this is a nonlinear one, of course. So we have discussed both descriptors on purpose because ADMET constants and partition coefficients, mostly log P, are used very widely. Of course, these are very simple descriptors and give us minimal information on molecular structure. Furthermore, in this article, very specific type of compounds was considered. So here, the applicability domain of this model will be really narrow. So on the applicability domain, I will say several words later. And we need to measure biological activity somehow to correlate it with molecular features. And let's discuss now what metrics of biological activity can be used in QSAR studies. And first, we will discuss what metrics of activity are used generally. So at the end, I will clarify. I will make I will clarify a little bit. Should we use grams per kilogram, or should we modify these values a little bit? In fact, we do. So we can, if we consider toxicity, we can use median lethal dose LD50 and median toxic dose. So generally, median lethal dose is measured in micrograms or nanograms per kilograms of body mass. So this parameter gives us dose killing half the members of a test set after a specified time. And here I would like to make a reference on the table on Wikipedia, because this, in fact, is very illustrative. What do I mean here? So, in fact, many compounds that we consider as essential for life can be lethal. For example, we can consider toxicity in rats, and we can consider oral toxicity. So there is some value that is associated. So there is some amount of water that can be toxic to rats, for example, that is when introduced orally. Or we can speak of sugar or glucose toxicity. However, the corresponding values are really, really high. The lower the value of LD50, the higher is the higher toxicity we have. And if we speak of LD50, we should always, we must always clarify toxicity on what organism and what type of toxicity, may it be oral or intravenous toxicity. So we can also consider median toxic dose, which is the dose at which specified toxic effect occurs in half the members of a test set. And we, on the contrary, can speak of therapeutic effect. And in this case, analogously, we can speak of median effective dose that produces a quantal effect, all or nothing, in half of the test set. And we can combine toxicity and effective dose in the so-called therapeutic index. So this metric is highly relevant for drugs with high non-specific toxicity. And this may be the case for anti-cancer drugs, for example, for chemotherapy. Many chemotherapy drugs are very toxic. However, we are interested in the ratio of effective dose to the toxicity. Because so when it's non-eligible, we should consider this value. And also, we should mention some quantitative metrics of inhibition activity. Often in QSAR studies, people discuss the so-called maximal inhibitory concentration, So for antagonist drugs, it's the concentration at which the compound blocks the activity by 50%. And usually logarithmic scale is used for a simple reason, because if we consider very active compounds, then concentrations may vary in very vary very highly. So concentrations may vary by powers of 10. And also we should mention affinity of the inhibitor. So this is basically the equilibrium binding constant of a ligand. Here, and also, as I've told you, we should always specify what type of assay we use to measure activity, in which organisms we measure it. For example, and we can speak here of activity in vitro and in vivo. Many QSAR studies consider activity in vitro for a simple reason. If we want to get some data on in vivo activity, we need to overcome many ethical examinations regarding the study, and data in vivo are really, really expensive to obtain. This is especially true in case of toxicity studies. So for a simple reason, for an obvious reason. And we need to mention how can we measure activity. So there are several protocols according to which we measure activity, and the most popular one is the so-called MTT assay. And in this type of assay, this tetrazole compound is used, which can be reduced to a formazine compound in a living cell. So if we measure toxicity to cells, for example, then we can use this type of assay to in this type of assay, we will measure the activity by this reaction. So this compound, this formazine compound, is highly is vividly colored, and it's purple. So moving from left to right, we get moving from left to right, there are more and more living cells that perform this reaction. And we can go back a little bit to discuss half maximal inhibitory concentration. So we, for example, can be interested in half maximal growth in inhibitory concentration. As for example, in the data set that we have discussed in the previous lesson. So then we discussed the toxicity to tetrachymena organisms, and we used IGC 50 exactly. A little bit, so let's discuss the relation between the binding constant and biological activity a little bit. First of all, we need to consider the famous lock and key model in which a model enzyme interacts with a model substrate. So the substrate binds to the enzyme, and in this complex, some structural, the structure of the substrate is changed. So after that, the product is released, and the key feature here, the key here is that the substrate and the enzyme need to have lock and key correspondence, no pun intended. So these should correspond structurally, and the better they correspond to each other, the more active process can be. Here we can consider the binding constant because it's basically the process of binding. And as I've told you about, we can establish linear free energy correlations, linear free energy relations to predict the binding constant of the system under study. However, I should mention that outside of the pocket, this geometry may be totally unstable. So what the enzyme does is that it stabilizes this geometry that favors the process, facilitates the process. And here I should also mention that this binding is usually non-covalent. There are some poisons, and there are some drugs, in fact, that bind to targets, that bind to a target covalently. However, if we speak of covalent binding, usually the unbinding cannot proceed. And usually covalent binding is associated with highly toxic effect. So considering covalent binding, we can mention the toxicity of carbon monoxide. For example, here there is human hemoglobin structure, and you can note here heme molecules, and here I've depicted the structure of heme with the carbon monoxide ligand. So basically, carbon monoxide binds to the iron atom strongly, and it prevents oxygen binding to this fragment, to iron, and this way it causes. This is the basis for the toxic effect. So, if we should also mention the so-called ADMET strategy, ADMET. Sometimes we can also include liberation in in our considerations. So basically, here we consider several the spectrum of properties of a given compound that are related to liberation of this compound in a living body, its absorption by cells of this body, its metabolism, and distribution in the body, and also excretion and toxicity are important. Here, I mentioned this only briefly because ADMET strategy is central to many drug development processes. However, ADMET studies are usually performed by using proprietary software that is basically a collection of many QSAR models that predict absorption, distribution, toxic, etc. properties of a given molecule. There are some tools for ADMET profiling, and you can refer to this article. You can refer to this review article for ADMET, for the tools that are open source. So in this ADMET, during ADMET profiling, researchers evaluate properties related to may evaluate properties related to solubility, for example. And as I've told you, solubility is a key property for a number of reasons. Because it's drugs or compounds activity can be related to how easily it penetrates cell membrane, and water-soluble molecules can penetrate, for example, via a channel protein, while fat-soluble molecules can potentially penetrate cell membrane directly. However, if we if a given molecule is too hydrophobic, then it can be problematic to transport it via body tissues, body liquids. On the other hand, if a given molecule is very hydrophilic, it may not necessarily penetrate the channel. That's why there are a number of rules to evaluate drug likeness in which potential solubility is in which solubility is the central property. So the first mnemonic rule is the Lipinski's rule of 5. So the terms are multiples of 5, and potential drug candidate should have no more than 5 hydrogen bond donors and no more than 10 hydrogen bond acceptors. So it should not be too water soluble. It should not be too lipophobic. It should not be. The molecule shouldn't be very large, let's say. So a molecular weight should be less than 50 daltons, and the octanol water partition coefficient should not exceed 5. So it should not be too lipophilic nor too lipophobic. Next, we should also mention the so-called goose filter. It differs a little bit, however, it also deals with partition coefficient, so it selects a more precise interval. It states some constraints on molecular refractivity and molecular weight. So there are some similarity. Speaking of molar refractivity, I've misspelled. It's related to molecular polarizability, and you can refer to Wikipedia on measurements of molar refractivity. So it's the property that was measured very actively in the previous decades. And you can also refer to the original publication on the so-called goose filter. However, nowadays, drug candidates and drug molecules that reach market differ from those that were considered as drug candidates or were released to the market in the previous years. So new drug molecules are generally larger, lipophilic, and polar. So these can be either this or that. And that's why researchers propose new metrics of drug likeness, as for example MCE18. So this MCE18 metric deals with several properties. So you can refer to the original publication, and it's this one. And if we consider drugs that were released to the market, then we see that gradually the structure, the molecule is larger and is more complex. So that's why MCE considers heteroaromatic rings in a drug candidate, aliphatic rings, chiral centers, and spiral points. So let me see, like an example of spiral point is structure like this. So fragment like this. So there are two cycles bound by one carbon atom. So MCE includes the portion of sp3 hybridized carbons, cyclic carbons, and also it includes the so-called normalized quadratic index. It's a topological index that you can so you can refer to this article I've sent you. Basically, it's indicates molecular shape. So it indicates if the molecule is linear or it's highly fused. So you can find the exact formula. However, as long as we are going to discuss this topological index next, so we are not particularly interested in the formula. So researchers made the authors collected statistics on drugs on new ligands, and in fact they found that many new ligands comply with this MC18 metric. So that was that's all on drug likeness, and let's discuss some QSAR approaches that can be used. So we have discussed descriptors that can be used in a QSAR study previously, and we can consider 2D descriptors, so we can represent molecules as molecular graphs and build connectivity matrices or distance matrices, or we can consider molecules as 3D objects. And the classic approach of the second kind is the so-called CoMFA, which stands for comparative molecular field analysis. In this method, basically, 3D structures of molecules of biologically active or inactive molecules are put into a lattice, and in points of this lattice, molecular fields are computed. First, energies of the van der Waals potential can be computed. The Lennard-Jones potential I've told you about can be used, and electrostatic interactions are computed. So we can use a probe molecule as, for example, a carbon atom with atomic charge +1, the probe atom the authors of this classic study used, and they put this probe atom in points of this lattice and computed values of these two potentials. So this basically, this approach is based on molecular force fields. It's related to molecular mechanics, and we can compose a matrix of computed molecular fields and use it for use it in a machine learning model. And here you can refer to the original article I've sent you. So what should be mentioned, that classic in this classic article, they correlated binding affinity of steroids to carrier proteins. This is important, of course. However, we should note that all molecules under study are structurally rigid. So this steroid core is composed of aliphatic, fused aliphatic cycles that are structurally rigid, and only side groups can show some conformational mobility, let's say. So in this figure, for example, you can see that it's quite easy to superimpose every molecule in the data set, and it's easy to orient them in the same way. So it's crucial to orient them similarly, orient the molecules similarly to obtain meaningful results. However, it's well, many biologically active molecules are structurally soft, so they undergo rapid conformational interconversions, and these can proceed really rapidly. However, this is a viable classic approach for structurally rigid molecules. So tomorrow we are going to have some practice. We will compute some additional descriptors, and we are going to use descriptors computed on the data sets we have discussed previously, and we will apply some machine learning algorithms. But today we should also mention in which situations QSAR models can be used. So here we should mention the concept of virtual screening. We have discussed it before because it's a very fundamental paradigm in chemistry, in computational chemistry. So if we have some chemical library that includes a great number of molecules, we can perform virtual screening by applying several filters. For example, we can use empirical rules. We can apply empirical rules like Lipinski's rule of 5 or Goose filter or MCE 18. And next we can filter molecules by chemical similarity. This can be 10-emotive similarity computed on molecular fingerprints, for example, the approach we have discussed previously. Then we can apply QSAR-based filters. And this stage may include many QSAR models that are applied consequentially. So many ADMET profiling studies use a great number of QSAR models. And we should also assess synthesizability of molecules from a virtual library, if this one is some kind of a virtual library. So we, by doing all this, we arrive at number of potential hits that can be used in experimental assays, for example. And if we speak of if we speak of a laboratory that applies QSAR to drug development, there is a closed loop workflow that is often used. So researchers can combine several data sources, may it be electronic databases, lab notebooks. Don't underestimate this, because inactivity data are very important, and they can also extract data from available scholarly literature. So this can be combined in a data collection that should always be curated for a number of reasons, some of which I will mention later. After that, the data repository is composed, from which we can extract data sets for QSAR study. So we obtain QSAR models that can be used for virtual screening. And next, experimental validation is essential, of course. So I've told you about the concept of applicability domain, and this is in fact central to QSAR, because to the field of to the QSAR field, because if we establish a model, it's always associated with a specific type of biological activity, and usually the dataset used is limited to some classes of compounds, and these two limit the applicability domain within which the model is reliable. So you cannot use a QSAR model, like, for example, this one, for molecules that are out of the applicability domain. So we cannot use structurally flexible molecules like chain, like lipids, for example. And there are some problems related to many QSAR studies, and the authors of this article highlight 21 typical problem, and I've extracted some really some relevant ones from this set. So if we compose a data set, we should always mind possible data heterogeneity. So we should not combine data on activity in mice and in rats, or activity in vivo and in vitro. And in fact, we should always try to use data from maximally similar assay protocols. And I've told you that units are key. So if we speak of toxicity, for example, milligrams per kilogram or nanograms per kilogram are usually present in a database. However, we should use smaller units because we are interested in activity per molecule, let's say. So molecular mass can vary greatly within a data set. So we should use this, not this. And sometimes there are collinear descriptors that degrade the quality of machine learning models. Usually, it can be this problem can be overcome by using the standard approaches, and this can be a problem only due to due to sloppiness, and sometimes people overfit models. So this overfitting should be avoided as well. However, what's important is that descriptors should be adequate. So we should select adequate descriptors that are interpretable and physically meaningful. And that's simply because mechanistic, every QSAR study ideally should give a mechanistic interpretation of biological activity that is predicted. Some, as for example, some compounds in a wide data set can be toxic via one mechanism, and some compounds can be toxic via the second. So we should not combine them, and this is the key part of dataset construction in QSAR studies. Sometimes we need to perform autoscaling, of course, and should limit number of descriptors. So now I would like to discuss the related field of chemometrics that I've mentioned in the previous lesson on linear free energy relationships. And but before that, let's consider the phenomenon of taste. So as you may know, taste is a complex phenomenon because it's a sum of receptor signals, and the same is true for smell. So correlations of models are models that predict taste or smell are really may be really complex. So sometimes it takes deep learning approach. It takes deep learning approaches to build a really predictive model, as was demonstrated recently. And the classic approach to predict taste may be a classic way to predict taste may be chemometrics. So what alternative to chemometrics is? We can use a panel of degustators, so a set of people that taste or smell some compounds or some mixtures, and document their observations. However, such an approach is prone to is problematic, really. So there can be high dispersion associated due with the subjectivity of taste perception or smell perception. So to overcome this, we can use we can use a specific device that combines ion selective electrodes. First, let's discuss on what ion selective electrode is. So here, the key part is of the so-called ion selective membrane. So this electrode is can be coupled to a reference electrode, and electromotive force of this system in this may be measured in this system upon the change of activity or concentration of substance under study in the sample solution. So this membrane is consists of several components. So the polymer is the matrix that keeps it all together. We need to include a plasticizer, and there are ligands in this membrane that interact with anions or cations selectively, bind them, and transport them to the inner filling solution. So depending on this ligand, this membrane can be penetrated by one ion or compound or neutral compound or another. And we can combine these potentiometric sensors in an array. So we can use a digital voltmeter and include a reference electrode. So having this array, we can calibrate it and construct a so-called electronic tongue. So each sensor in this array is basically some kind of descriptor. If it's calibrated to a particular compound, we can study complex mixtures like body liquids, some natural oils, or even beverages. So this is the schematic representation of electronic tongue. In real implementations of these real implementations look like this. So indeed, there are arrays of sensors that can be put into a sample solution, and we can use sample solutions. We can make a set of sample solutions that will help us to calibrate this array. So basically, we built a machine learning model that correlates signals from each sensor to the known concentrations. And such a device can be used to study many types of samples. So it can be usually electronic tones are practice-oriented, application-oriented. So an electronic tone can be used to study tomato taste, or it can be used to study sunflower oils, or it can be used even to study beer. Like in this article, for example, the team of researchers studied different beers like this, and they measured even bitterness of beer, which is quite complex quite a complex property to measure if we use a device and not a panel of degustators. So before finishing this lecture, I would like to mention a couple of online resources that can be really helpful. First of all, I should mention the QSAR DB. It's a site in which datasets and QSAR models are deposited. So you can download data from this site, and we are going to discuss several data sets that are deposited here. So I provide you a link, the link on this site, and so you can look and try. I suggest you to do that. We are going to use some data sets from this site tomorrow. And also, for those of you who have never heard about this community, I suggest you following this community in the VK social network, because here you can find a lot of textbooks and info on cheminformatics, on QSAR studies, and molecular modeling. So this is all for today.